Hello, everyone, and welcome to today's webinar. Getting to the heart of the matter, novel analytical technologies provide a new perspective on a complex organ. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Curiox. To learn more, please visit curiox.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speaker, Dr. Rebekah Gundry, Professor and Interim Chair of Cellular and Integrative Physiology and Director of the Cardiomics Program at the University of Nebraska Medical Center. Rebekah, you may now begin your presentation.
Thank you. All right, you should be able to see it. There we are. Okay, thank you so much for the introduction and the opportunity to be here today. I am really pleased to tell you about some of the new approaches that we're developing to learn more about the human heart. And as I go along, I'll tell you a little bit about how the Curiox laminar wash technology has really transformed some of our workflows to allow us to achieve some of our specific goals. So my journey to the heart really started back when I was in graduate school and postdoc. And as a grad student, I gained a lot of experience in mass spectrometry analysis of biological molecules. And so when I started my postdoc, I was really interested in gaining skills in pluripotent stem cell technology because of the possibility that it held for the future of cardiac research. And so when I started with embryonic stem cells, but soon after I started my postdoc, the new iPS technology emerged. So induced pluripotent stem cells. And so this technological breakthrough really fueled my excitement about stem cell technology even more. And so the term pluripotent stem cell refers to the ability of these cells to self-renew and differentiate into any cell type in the human body. And there are two types of pluripotent cell. They include embryonic stem cells, which are generated from the inner cell mass of an embryo, and induced pluripotent stem cells, which are reprogrammed from somatic cells. So embryonic and induced pluripotent cells have the same potential, but with iPS technology came new possibilities. Not only can we avoid using human embryos, but we could potentially make these from any patient with the disease. So it was now possible to think that we could model a disease, test drugs to find the best therapy for a specific patient, or maybe even repair damaged tissue. So I was really intrigued by the kind of what if question, if we could use iPS technology to better understand the heart and heart disease. And so as most postdocs do, I thought a lot about what is it that I could do to have an impact on the field. And that for me became obvious fairly quickly, because although we could pretty easily convert pluripotent stem cells into a dish of spontaneously contracting cells like you see here, the cultures are generally heterogeneous. And if this heterogeneity is variable and ill-defined, it can impact the interpretation of any functional data that's generated. And while we could make beating cells that express some cardiomyocyte markers, this is really not the same thing as saying that we generated a very specific cell type. And depending on the question, we think that you probably need to have different cell types to be relevant. Like, do you need a right atrial cell or do you need a left ventricle cell to do the disease modeling or the drug testing that you want to do? So the enthusiasm regarding the potential of iPS technology is tempered somewhat by these really major technical hurdles. And so we and others had the idea that we could use immunophenotyping to solve the problem. So immunophenotyping is a strategy where panels of antibodies to surface proteins is used to identify and isolate cells with a defined functional phenotype. So this approach is well established for isolating long-term repopulating hematopoietic stem cells for transplant. And it's a clinically relevant strategy because it does not require genetic editing, and it provides a way to standardize how cells are evaluated and compared among labs, protocols, and applications. And we thought that this could really benefit the pluripotent stem cell-derived cardiomyocyte field if we could develop marker panels to distinguish different cell types and different maturation stages. However, we don't yet have enough information about what proteins sit on the cell surface in the pluripotent stem cell system, as it's much less well characterized than the hematopoietic stem cell system. So while some researchers approached this with an antibody-based screening approach, I knew that we could exploit our expertise in mass spectrometry to do this more efficiently. So mass spectrometry is a technology that allows us to simultaneously identify, quantify, and characterize biological molecules without having to rely on antibodies. And so while antibody-based screening is limited to really a relatively few number of really high-quality antibodies, and it has limited multiplexing capabilities, mass spectrometry, on the other hand, can identify and quantify hundreds to thousands of proteins in a single experiment without antibodies. And so we used mass spectrometry to create the first baseline views of pluripotent stem cells, so the cell surface of pluripotent stem cells in both mouse and human systems. And from these data, we developed markers for selecting pluripotent stem cells from heterogeneous populations. And then we went on to use that information to select inhibitors to a cell surface protein GLUT1 that allowed us to selectively eliminate pluripotent cells from their derivatives. But as we continued testing, we found that one of the small molecules that we were using does not actually inhibit GLUT1, although that was its reported mechanism of action. Instead, we found that it inhibits an enzyme called NAMPT, which is a rate-limiting enzyme in one of the NAD salvage pathways. So from there, we defined how and when stem cells differentiate to cardiomyocytes, they become increasingly metabolically flexible. And then this discovery ultimately led us to develop a method to eliminate tumorigenic cells from heterogeneous cultures of stem cell derivatives, making them one step closer to being useful for drug testing, disease modeling, and regeneration. However, as we continue to generate more views of what these cells in a dish look like, we increasingly began questioning, how can we really know that we have the right markers? How do we avoid those markers that are just an artifact of the fact that we're culturing cells in a dish? And we started to think about this because of the only cell type that we ever use for this discovery process are those cells that we make in a dish, then we're only ever going to be able to see the proteins that the cultured cells make. But we already know ahead of time that the cells in the dish don't actually mimic the myocytes that we find in the adult human heart. They're structurally and functionally immature. So then we started to think about, well, if we're only looking at the cells in a dish, we're probably in a situation that's kind of like this cartoon of the lamppost problem, that it's likely that we're not even looking in the right place if the goal is to make sure that we have markers for cells that most closely mimic those that we find in the adult heart. So we started thinking about how much better would it be if we could compare real heart cells to those that we make in a dish? Because we thought that if we could look at the primary cells, this should tell us exactly what should be there for specific cell types, for specific regions, for the adult maturation stage. But the problem was at that time, we didn't have a way to do it. We didn't have, the technology was not good enough. We didn't have a way to isolate cardiomyocytes from human hearts. And even if we could isolate them, we could never get enough material that we needed to do that cell surface profiling by mass spectrometry method. We typically need 50 to 100 million cells, and that's way more than we thought we would be able to get from human hearts. So we have this thought about, wouldn't it be really great to look at primary cells, but our tech isn't good enough. And so really around the same time we were kind of struggling with this, I started to have some informal conversations with a clinician friend of mine who takes care of patients in advanced heart failure. So heart failure is a condition where the heart doesn't pump enough blood to meet the body's demands. And once a patient is in heart failure, the first line treatment is pharmacotherapy. But approximately 10% of patients will fail to respond to medication alone and go on to develop advanced heart failure, at which point they can receive a heart transplant or mechanical circulatory support. However, hearts are available in only limited supply. Support devices generally have variable patient outcomes, and while current drugs target issues that are secondary to the failing heart, like neural hormonal activation and renal function, they don't directly prevent the structural molecular abnormalities that are associated with heart failure. So there's currently a need to increase the supply of donor hearts, find ways to predict patient outcomes, and hopefully develop therapies that could directly address the structural defects in the heart. So as me and my friend kept talking, we increasingly focused on how we really just don't know enough about the molecular detail of the healthy or the failing human heart. And while we do know that cell surface proteins like beta-adrenergic receptors, calcium, sodium, and potassium channels, and connexins are required for normal heart function, we just don't have a complete picture of what's really present in different cell types in the human heart and how or if it changes with disease. And so since our long-term goals really include, can we find better drug targets? Can we find better biomarkers? It made a lot of sense to us that we could apply our expertise in studying the cell surface proteome to start filling in these gaps. And why do we believe that? Well, we know that cell surface proteins can be used for immunophenotyping, as I told you about for stem cells, but they're so much more useful and informative than that. So cell surface proteins play critical roles in maintaining cell structure and signaling. So we expect that if we had a better view of the proteins that sit on the cell surface and how they change in disease, we can learn a lot about how cells respond to their environment to maintain normal cardiac function, how they respond to stress and impact disease, and we can find drug targets. And because cell surface proteins can be cleaved or shed into the circulation, they can be exploited as remote sensing biomarkers of disease. All of these applications, in theory, would be relevant to addressing some of the clinical needs that my clinical friend has in treating his patients with advanced heart failure. So here we are again, kind of back at this. Wouldn't it be really great to be able to look at cell surface proteins on human heart cells to address some clinically related questions? But again, the technology wasn't there. So we had this need from both the stem cell side and the heart failure side. And it was really just frustrating that we did not have a technology at the time that would allow us to do this. So again, the major issues were that we didn't have a way to routinely obtain heart tissue to isolate cardiac cells. And the approach that we use for discovering cell surface proteins requires a lot more starting material than we expected that we could ever get routinely from human heart. But it was really exciting to think about what would be possible if we could do it. And we were really propelled by this quote from theoretical physicist Freeman Dyson that states, that says that new directions in science are launched by new tools much more often than by new concepts. And the effect of a concept-driven revolution is to explain old things in a new way. The effect of a tool-driven revolution is to discover new things that have to be explained. So consider what the Lumiere technology did for revealing previously unknown history of the Mona Lisa. So the Mona Lisa was painted in the 1500s, but it wasn't until the Lumiere technology that was applied in the 2000s did researchers discover that the Mona Lisa was generated using the Spolvero technique, which is where the drawing is transferred from to the canvas with charcoal. So it's originally drawn on paper first. So this raises the fascinating possibility that somewhere out there is a paper drawing of the Mona Lisa by Leonardo that may still exist. Who knows? So back to science. So just like art, really every aspect of what we do on a daily basis in a biomedical research lab depends on the technology that we have available to us, what we can measure, what we can see, whether it's a microscope, whether it's a CRISPR technology. Our ability to do what we do depends on the technology we have available. And sometimes if we want to learn new things, we have to develop new tools. So we set out to develop a platform that would allow us to discover proteins on the cell surface of human heart cells. And to do this, we needed to develop 4 key components. One, a way to obtain the tissue, a way to get the cells out of the tissue, a way to profile the cells, and a way to analyze the data efficiently. And so we started by working on the third component, which was to develop a surface protein profiling method that will allow us to specifically identify cell surface proteins from low amounts of starting material and something that would be somewhat automated to make it more reproducible among users. And so for this, we started with the classic cell surface capture method that has worked well for us for all of the stem cell work that we had done today. So briefly, the method works by biotinylating glycans on cell surface glycoproteins of intact cells. And after digestion, biotinylated glycopeptides are enriched by streptavidin beads, and then the deglycosylated peptides are released by PNGSF and then analyzed by mass spectrometry. So our classic method was all manual. Everything was done in tubes. There was a lot of sample losses throughout the process, very laborious 4-day process, really not practical for primary heart cells. So we improved this by adding a chemical catalyst to maximize labeling at the start, implementing automated robotics along with our recently described avidity assay for quality control of the streptavidin beads. And then we also developed a cleanup method that efficiently removes detergents so we can use harsh detergents during the bead washing upstream to minimize the non-specific binders. And so altogether, this new method is done in less than half the time. So only 2 days, we can use a lot lower starting material and still achieve really good coverage of the surface proteome. So this microscale cell surface capture works well for a range of cell types, and whether we start with 200,000 or 2 million cells, we're detecting 300 to 700 proteins in a single experiment with greater than 80% specificity of capture. So overall, the approach is working well for sample sizes that we expect to obtain from human hearts. And importantly, beyond just protein identity, the method allows us to determine sites of N-glycosylation and extracellular exposed peptides. And this information helps us to confirm orientation of the protein in the membrane, as many cell surface proteins are not completely characterized yet. And this information is really critical when you need to develop antibodies to target the extracellular domain. So the next piece of the puzzle on the analytical side was a set of bioinformatic tools that would allow us to process the data in a standardized and transparent way. And so over the past few years, we've developed a suite of bioinformatic tools for rapidly going from our mass spectrometry data to prioritize and annotated lists of candidate cell surface markers. So Veneer and Surface are the tools that we use to ensure that we process the data in a standardized manner. And Surface Genie and Vision are what we use to rapidly prioritize candidates for downstream applications. And I don't have time to tell you about these in detail. You can ask me in the Q&A session, or you can check them out there on our cell server website. But I will show you some examples of their utility as I proceed through the talk. So overall, with these new tools, we have the ability to process large amounts of data in less than just a few minutes, compared to previously what would have taken us weeks to months to process. So at this point, we had really the technical and the bioinformatic approaches sort of figured out, but we didn't really have an ideal strategy for regularly obtaining human tissue. So fast forward to the spring of 2019, when I was interviewing at the University of Nebraska Medical Center in Omaha, and I met some amazing clinicians and surgeons, and pretty quickly, within a few minutes of talking to them, I really knew that UNMC would be a great new home for us, as it would provide us the opportunity to do some things that we couldn't really do at my previous institution. And I'm really glad that we moved, because we were able to finally get to where we wanted to be, which was to develop the human heart tissue strategy that we needed. And so for this, we needed a practical method that kind of minimized the amount of time from the operating room to processing and storage in the lab to maximize tissue integrity. We needed a tissue collection strategy that would be deployable on demand, as it's really common for there to be just really limited advance notice that a surgery is going to happen, and it's not uncommon for these surgeries to occur at night. And we wanted to be able to process the tissues in a way that would be amenable to lots of different types of analyses, not just cell surface proteomics. So long story short, we worked with the clinicians to develop a standardized workflow where, as soon as the OR is scheduled, our lab delivers some supply packs to the operating room. And then the surgeons place the heart into preservation solution immediately after removal from the donor. And then they do some crude dissections for us into 11 different regions of the heart. And then we bring that back to the lab and further process it and store it either as cryopreserved tissue, as well as a number of other sample formats. And so we use a similar process for transplants, LVADs, and rapid autopsy. And since starting this collection workflow just 2 years ago, basically soon after I moved to UNMC, we've already collected more than 1,500 cryopreserved aliquots from more than 150 donors, and this doesn't count all the other specimen formats. All right, so now that we have this increased access to tissue, we can fully optimize our method to isolate the heart cells in a way that preserves plasma membrane proteins. And so we have methods for both cardiac fibroblasts as well as cardiac myocytes, but today I'm just going to talk to you about the cardiac myocytes. So when we think about how do we isolate cells from the heart, the devil really is in the details. There's a lot of technical challenges to working with human hearts. So there's often, even having direct access to them from the OR, there's still limited amount of tissue per donor, because the hearts still need to go to pathology for full evaluation. The heart is, of course, a very dense tissue with multiple different cell types. Unlike rodents or animal models, we can't just perfuse the whole heart with digestion enzymes. We have to start with kind of chunks of tissue. We can't use enzymes like trypsin or trypsin-like enzymes or collagenases that contain trypsin-like activities, because what do? Well, they cleave off all the things we're interested in. They cleave off our cell surface proteins. The amount of fibrosis can vary depending on the disease state. Cardiomyocytes in particular are very sensitive to centrifugation and shear force. methods for isolating cells from frozen tissue was not really well developed before. And once you isolate those cardiac myocytes from the human heart, they don't replicate. It's not like other cell types where you can expand them after you isolate them. So really what you isolate is what you get. So long story short, the human heart tissue contains, multiple cell types like myocytes, cardiac fibroblasts, immune cells, and endothelial cells that all work together to maintain cardiac function. So our goal was, how do we get the cardiomyocytes out and away from all the other cell types in a way that just doesn't destroy the cell surface proteome? So what we do ended up developing as a method that we start with cryopreserved tissue. We use a tissue slicer to create consistently thin slices of tissue. And then we digest with collagenase and DNase to disrupt that extracellular matrix, but otherwise leave the other plasma membrane proteins alone. And then in our previous version, we had used a Percol gradient to try to enrich the cardiomyocytes and separate out debris. And then as just kind of a final step, we used negative selection to remove immune cells and endothelial cells. And so we end up with cardiomyocytes. But when we learned about the laminar wash technology available from the Curiox, we thought, oh my gosh, wouldn't this be amazing if we could get rid of that Percol gradient? Percol gradient is such a pain to do, and it doesn't work that well, and there's still a ton of debris left behind. So after a few quick tests, it was pretty clear pretty quickly that this was going to be a game changer for us. So now our new and improved version uses the laminar wash instead of the Percol gradient. We still use the negative selection at the end just as a safety net, but generally the Curiox is really great at getting rid of all the kind of non-cardiomyocytes and the debris. And so just some crude pictures. See, the Percol gradient, sure, it works, but the cells are definitely not happy. One, it takes 8 hours. The cells, lots of centrifugation steps. The cells are really stressed. There's a lot of debris. Compared to the Curiox wash, now it's down to just 4 hours. The total harvesting process is 4 hours, not the wash. The cells are a lot less stressed, a lot less debris, and we get a lot higher yield. So this is something that really helps us to be able to have lots of people in the laboratory be successful at this process because it's a standardized approach now. All right. So combining the microscale cell surface capture method with a new bioinformatics tools is what we call our cell surfer platform. And it's applicable to any mammalian cell type, but our main goal originally was to make it work for human heart cells. So that's what I'm going to tell you about. So applying the cell surfer platform to the 4 cell types that we isolate from human hearts, we identified more than 1,100 cell surface and glycoproteins to create the first experimentally defined map of the proteins that sit on the cell surface of different human heart cells. And as expected, we find a lot of transporters, G protein coupled receptors, drug targets, and ion channels. And so these data are a rich resource for many applications, from basics about protein sequence to disease to insight to biomarkers. And I will show you a few examples for each of these applications. So to begin, I'm going to show you why the details we learn in our experimental data are really important, as many cell surface proteins are not completely or accurately characterized yet. So our bioinformatic tool Veneer generates a really highly curated output of surface exposed glycopeptides. And so comparing our data to several broadly used public repositories and data sets allows us to see why predictions and annotations alone are just not enough when we're doing surface proteomic studies. So for 30 proteins, our experimental data are inconsistent with the transmembrane topology that's described or predicted in UniProt. For 66 proteins, they're not predicted to be N-glycoproteins in UniProt. 358 are not annotated as plasma membrane or secreted in the Human Protein Atlas. 249 are not contained in one of the largest human cardiac tissue proteomic studies published a few years ago. 327 are not in our own proteomic analysis of whole cardiomyocytes. 155 of the cardiomyocyte proteins are not contained in the largest to date single nuclei RNA-seq study of the human heart, and 422 are not contained in the predicted the human surfaceome. So this just reminds us that we cannot accurately predict which proteins are on the surface of the cell. We really need to measure them. And if we want to measure them, we have to use an approach that specifically enriches for them, as whole tissue or cell lysate analyses are not sufficient. So now I will move on to how we're using the data to identify potential new markers of specific cell types in different regions of the heart. And then with that, we expect that will inform future drug delivery and antibody development. So here I'm showing an upset plot that illustrates the overlap in identifications of proteins that we observed among different cell types. So for us, kind of easy, most interesting are those proteins that we observed only in a single cell type. So of those, we observed some proteins only in cardiomyocytes, and these include CACNA1C and SCN5A. And similarly, in cardiac fibroblasts, we only identified CILP and ECM1 in cardiac fibroblasts. And also for endothelial cells and smooth muscle cells, we also had proteins that we observed only in a single cell type. So these are all examples of proteins that are otherwise known and expected to be in these specific cell types and provide evidence that the overall strategy is working and that there's a lot of possibilities in these data for developing cell type specific markers for a variety of applications. And so one of these new proteins is LSMEM2, which is a protein that we only identified in the cardiac myocytes. So LSMEM2 is leucine-rich single-pass membrane protein 2, and this emerged as an interesting protein because our lab has done thousands of cell surface capture experiments. And so far, we've only ever identified LSMEM2 on the surface of cardiomyocytes we isolate from human heart or stem cell-derived cardiomyocytes. It's a protein for which there are no publications, no known function, no previous observations in the heart. UniProt does not predict it to be a plasma membrane protein or glycoprotein. And so using our surface tool, we can easily see that different algorithms for predicting transmembrane domains provide different results regarding which portion of this protein is actually in the extracellular domain. And this discrepancy is probably due to the fact that this protein doesn't have a canonical signal peptide, which is one of the things that many of the transmembrane algorithms require. So this might explain why our experimental data conflict with some of the UniProt predictions. So there were no monoclonal antibodies available to LSMEM2 when we started. So remember, the microscale cell surface capture provides experimental evidence of the extracellular domain. And so we use that information to develop new monoclonal antibodies for targeting the extracellular region of this protein. And these antibodies are useful for imaging heart tissue as well as flow cytometry. Here I'll just show you some imaging where we see this protein only in cardiac myocytes. And the staining pattern is similar to that of ion channel SCN5A. And we see LSMEM2 in the intercalated discs where it has some overlap with connexin 43. So when we were browsing resources to see what's known about this protein, we found that the Human Protein Atlas shows some immunocytochemistry data with a polyclonal antibody that shows LSMEM2 is present in 3 different cell lines. So HeLa, Jurkat, and U2OS. All of these are not cardiomyocytes. This was interesting to us because our lab, as well as 3 other labs that I know that do really great surface proteomics, have never found LSMEM2 on these cell types. So it could be for lots of reasons. So we explored that a little bit. When we use our monoclonal antibody in flow cytometry analyses, we do not detect LSMEM2 on these non-cardiomyocyte cell types. And as a non-antibody-based method, we also use a SureQuant assay. So it's a targeted mass spectrometry assay where we program the mass spectrometer to specifically look for just LSMEM2. And when we do this, we find LSMEM2 in our LSMEM2 transfected HEC cells, as well as primary cardiomyocytes. But again, we do not detect any protein level evidence for LSMEM2 in HeLa, Jurkat, or U2OS. So these data just remind us for the need for continued vigilance when relying on predictions and antibody-based evidence for surface protein localization. All right, so ultimately, when we started this project, we had relied only on gene ontology or database annotations, we would have never selected LSMEM2 as a viable target. So the stringent criteria for classification as a surface protein based on our experimental evidence and streamlined analysis tools really helped us to not overlook this protein of interest. And LSMEM2 is just one example of several that we have found for which they're in the heart, but there's no previous reports they're in the heart and there's no known function. So beyond cell type restricted proteins, we're also interested in cardiomyocyte proteins that are region restricted, as they could be potentially useful for how do we know that we've made the right stem cell derivative for disease modeling or diseases that affect specific regions of the heart, like the atria or the ventricle. And then also, thinking about future delivery of payloads. And there might be a time where we want to deliver a drug specifically to the diseased left ventricle and spare, any treatment to the right ventricle, for example. So here I am again showing an upset plot that shows the distribution of proteins that we identified in cardiomyocytes from 5 different regions of the heart. And as expected, we have proteins like SCN5A and CAC-NA1C, where we find that in cardiomyocytes from all regions of the heart. And then we had other proteins that we only identified in the right and left ventricle, including TMEM63A and Visorin, and then BMP10 we only identified in the left and right atria. So these, again, are examples that are observations that are consistent with expectations. So this lends promise to the other potentially region-restricted candidates identified that are currently undergoing validation. Okay, so far I've shown examples of what we can learn about the fundamentals regarding cell surface proteins in the non-failing heart. And so now I will tell you about how we can use this approach to learn about heart failure. So in a small, rather pilot study, we analyzed cardiomyocytes that we isolated from failing and non-failing hearts. And we quantified 490 cell surface glycoproteins in this experiment. The PCA, principal component analysis, shows that by looking at the surface protein profile, the data cluster based on patient groups. So the heart failure patients cluster together and away from the non-failure patients, which suggests that differences in the surface proteome alone are enough to distinguish these patient groups. Using our Surface Genie tool, we started with a list of 490 cell surface glycoproteins, and what Surface Genie does is prioritize those things that are most able to distinguish between groups. In this case, less than a minute later, we have a rapidly prioritized list of proteins that are most likely able to distinguish between patient groups. And here I'm showing the rank order with some examples labeled based on our OmniGenie score that, again, is calculated by the Surface Genie tool. And those proteins in purple are those that are decreased in failure compared to non-failure, where the green are the proteins that are increased in failure. And again, this is just rank order prioritization of the things that are most likely to allow us to distinguish. And so this is really the first analysis of its kind. We don't really have, other great data sets that we can directly compare to, but we can, look at the proteins that we're seeing and evaluate if what we find is consistent with expectations versus new. And so just a couple of examples, we find that S1P receptor 3 is decreasing on the surface of failing cardiomyocytes. And this protein has a role in protecting the heart from ischemia reperfusion injury. ENT-PD1 is increasing in failing myocytes, and this protein has a role in regulating the extracellular nucleotide levels, including adenosine, which has cardioprotective effects. So other proteins that we find that are changing have been, some of them have been broadly linked to cardiovascular disease, but not yet specifically linked to the cardiac myocyte and or heart failure. And then for some proteins, there's no link at all to the heart previously, and this includes LSMEM2. So far, the function of LSMEM2 is not defined, so we don't know why it's decreasing in heart failure, nor if it's a drug target, but we're working on it. So when we think about how do you know what a protein does, what's its protein function, there are a lot of pieces to this puzzle that need to be assembled. And so far we know the amino acid sequence, and by mass spectrometry have confirmed the site of N-glycosylation, which also tells us how the protein's oriented in the membrane. Evolutionarily, it's only present in animals with functionally 4-chambered hearts, so septated 3-chamber or 4-chambered animals, so it's not present in things like the zebrafish model, for example. It has some predicted disordered or low complexity regions, and the disordered structure and the late evolutionary emergence together, there's some evidence that this may correlate with the emergence of novel protein functions in higher order eukaryotes. We see it along the myocyte and in the intercalated disc, which is part of the cardiomyocyte that's critical for force, electrical, and chemical transmission between cells. And we see it in cardiomyocytes from all regions of the heart. So questions we're currently working to answer include determining what's the structure of the glycan on this protein. Within the intercalated disc, where is it specifically? Is it in the desmosome? Is it in the gap junction? And who does it talk to? Who does it bind to? Where is it, specifically, where is it shed and cleaved? What happens if there's no glycosylation? And what happens when there's too much or too little of this protein? So these are all ongoing studies. All right. So now I'm going to move on from looking at the changes we find in disease to show you an example of how we can use the data from the primary cells to answer our original question, which is how can we exploit primary cell data as a benchmark to make sure that we have the kinds of cardiomyocytes in a dish that we really want to have. And I'll just show you one example. Okay, so for this example, we compared surface protein profiles of stem cell-derived cardiomyocytes over time in culture. And then we also compared that information to what we see in primary cardiomyocytes. And again, as one example, we found the cell surface protein CD36 was observed in really low abundance in early differentiation in a dish, and very high in later stage cardiomyocytes in a dish, as well as in primary cardiomyocytes. So with our collaborator, Kimbo Healer, who did a lot of this functional work, we found that the CD36 high and CD36 low populations have distinct transcriptomes and functional potentials. And that the CD36 high sorted cells were overall more mature compared to the CD36 low, as evidenced by having more mitochondrial membrane potential, more mitochondrial content, and increased fatty acid uptake. So overall, the CD36 high versus CD36 low have a different functional potential. So what's the impact of this? How can we use that to address our original goals? Well, it has an impact in this example of how we can use stem cell-derived cardiomyocytes for drug testing. So you can read all the technical detail in the paper. I'm just going to give you the take-home message. But in short, doxorubicin-induced cardiotoxicity is a problematic side effect of cancer treatment. And so if you take doxorubicin and apply it to stem cell-derived cardiomyocytes or primary human heart cells, that will have a toxic effect. So there is a drug, dexrazoxane, which can protect against this toxicity. And while we see that it can protect against this toxicity in primary cells, traditional stem cell-derived cardiomyocyte models have not really adequately mimicked this response because it doesn't have this protective effect in vitro. But if you sort the cells, CD36 high versus low, now the CD36 high cells that are more mature actually can now replicate the effect, this protective effect that we see in vivo. So this is just one example of how we think about cell surface markers being useful for selecting cells that will be more useful for drug testing and disease modeling in the future. And besides CD36, we have other potential maturation markers. Some of these are even more restricted to later stage cells than CD36. So these include ASPN, LPL, CPVL, and VSIR. And we're just really in the process of evaluating the utility of these for selecting cells with perhaps more accurate functional properties. And to end, another area where we're pushing the approach and the technology and the applications is how do we exploit these data for developing remote sensing biomarkers? So you might be familiar with approaches for biomarker discovery that start by comparing, let's say, serum. Let's say serum samples across patients, patient group A versus patient group B. You look for what's different and you find things that are different, but then the challenge is the changes may not really be related to the underlying pathology or even your organ of interest. So it can be challenging to move those forward. Another strategy is to start with the organ and the pathology of interest and find what changes and then go from there to see what's actually present in the circulation. And so what does it have to do with cell surface proteins? Well, cell surface proteins are often shed or cleaved and detectable in the circulation. And if you have these proteins that are either organ or cell type specific, or let's say this cleavage or shedding activity is specific to a disease state, conceptually, these could be really valuable biomarkers. And without having to do all that screening, you can actually just develop antibody-based or mass spectrometry-based methods to specifically detect these proteins in the circulation in your conditions of interest. So as a couple of examples of why we're really thinking about this as a possibility for the future, consider one example, which is SEMA4D. So SEMA4D is a protein that we found its abundance on the surface of cardiomyocytes isolated from failing hearts is lower than the abundance on cardiomyocytes from non-failing hearts. And there have already been some studies published, and I'm only showing data for one of them, that found that the abundance of SEMA4D in the circulation increases in failure compared to non-failing. And so the fact that it's lower on the surface of the cells and more in the circulation could be because it's cleaved. And it has, there's some evidence to show that it can be cleaved by atom 17, maybe in platelets, but it also could happen in cardiomyocytes because Atom-17 is also in human heart cells. So we don't really know if this is necessarily a direct connection, but it just raised the possibility that there could be this connection there. And then knowing what is doing the cleaving and why it's cleaving could be certainly informative for drug targeting. And then also exciting is our LSMEM2, our new protein of interest. We also found to decrease on the surface of cardiomyocytes and heart failure. And we have some very preliminary dot blot shows that we can detect this protein in serum after a myocardial infarction. Now, of course, a myocardial infarction is not the same context as heart failure, but again, it just raises the possibility that LSMEM2 is perhaps shed or cleaved, or maybe it's in extracellular vesicles. So this is, these are just two examples that highlight the possibility of how we can exploit these surface protein data to develop clinically useful biomarkers. And so in summary, CellSurfer is a new enabling technology that allows us to view the heart with really an unprecedented level of molecular detail that's not accessible by transcriptomics or traditional proteomics approaches. And we've been able to overcome some previous limitations that we had on the technical scale. And so now we can apply this technology to advance towards our long-term goals that I told you about at the beginning that we have, certainly not yet fully answered, but at least now we have the tools to do it. So with stem cell work, we're well on our way to strategies that allow us to improve the ability to identify and isolate cells that are specific cell types, maturation stages, and regions, which we believe will improve their utility in drug testing and disease modeling. For the clinical work, we now have strategies to work with primary human cells and have identified cell type and region restricted targets that may be useful for directed payload delivery or revealing region-specific changes with disease. And of those proteins that are restricted to the heart or change really only in disease, at least we have a path forward to determine if we can detect them in the blood and exploit them as useful markers. So more broadly than just our own long-term goals, these technical capabilities and data make a bigger impact. So this is really the first experimentally derived map of surface proteins on various cell types in the human heart. And it includes the first protein level evidence for more than 70 proteins, including many that are not yet characterized. So the molecular detail that we provide regarding the glycocytes, the transmembrane orientation, and the cell type is really just the first basic steps you need for structure function studies that seek to determine what are the function of these yet unknown proteins. We have a lot of new cell type and region restricted candidates, and those cell type and region restricted candidates, along with the maturation stage markers, we think will be tools that anyone in the field could use to identify and characterize cells that they make in a dish. Knowing the proteins in the heart not only provides a rich resource for drug targets for cardiac diseases, but also for prioritizing targets for drugs for non-cardiac diseases. So if you want to develop a drug for cancer, it'd be really great to know that that is not going to likely have cardiotoxic effects. And so this, again, is just for prioritizing those drug targets that might be more safe. And finally, we're adding new molecular detail of what's happening during heart failure and unloading. And as we analyze now the larger cohorts that we have access to that we've been collecting over the past few years, we expect this will inform new strategies to target for therapies and biomarkers. And so the CellSurfer platform is overall applicable to any primary cell type. We expect that as we move forward and routinely profile primary human cells, we will be able to gain new insights into cell type specific views of physiology and disease. We can uncover drug and therapy payload targets without having to rely on problematic cell lines. And we should be able to reveal shed and cleave cell type specific proteins that can be explored as remote sensing markers. And so we have a lot of collaborations that are ongoing to do some of these things, from neural cells to liver cells, pancreatic islets to retinal cells. We're able to, for the first time, now that we have this miniaturized technology, define what's on the cell surface and how they differ in disease or developmental stage. And so already it's leading to some exciting new findings. So for the stem cell models of human disease, we have for spinal muscular atrophy, Ehlers-Danlos syndrome, and there's a GATA4 mutation that affects cardiac fibroblasts that we're learning new information. And then in cases where we have primary cells, the technology has provided some new gene therapy targets for macular degeneration and blood cancer. And also our work on the beta cells, so both rat, mouse, and human cells, developing new payload delivery and cell sorting markers for those. So overall, the value in being able to study primary human cells really motivates us to keep pushing the limits of this approach. And over the past year, Ronald, me, and Melinda in the lab have been working on the next generation of the technology, which we call nanoscale cell surface capture. And so here we're actually now using the laminar wash not just to isolate the cardiac myocytes, but it's really awesome for that first upstage part of the process where we have to biotinylate our cells, because again, we don't have to do any centrifugation. So we can do all of that process in a really small sample format right on a 96 well plate without centrifugation. So it really speeds things up, allows us to not lose as much material. We've also swapped to a simpler plate-based sample digestion method that can be done now in an hour instead of overnight. And we've onboarded a new fully customizable robot along with our Levitate platform of 3D printed accessories that allows us full control over liquid handling. All in all, just to really help us totally automate the process as much as possible, we can start with a lot less sample material. So now we can do the entire process in one day instead of two. We can start with fewer than 100,000 cells and still identify quite a number of proteins. So up next, we are now able to do some things that we haven't been able to do even with the microscale cell surface capture. So we're working to refine the temporal pattern of surface proteins in cardiomyocyte cultures with a lot greater temporal resolution, because now we don't need as many cells to do this. We're really curious of if we could exploit this technology for looking at exosomes or extracellular vesicles from cardiomyocytes, either in culture or primary cells. Now able to get, I think, to pacemaker cells as well as cells from atrial appendages. And we can now define with greater resolution what's different among the chambers and non-failing versus failing hearts. Now that we have a lot larger sample cohort than we had before. And now that the sample handling is well optimized, we're working on changing the chemistry at the front end so we can expand our view of the surfaceome to more than just the N-glycosylated proteins, but also proteins that are exclusively O-glycosylated as well as those that are not glycosylated. And so with that, I thank all the amazing past and present lab members for their creativity and pushing these technologies and applications forward. Our amazing collaborators, funding sources. Thank you for your attention, and thanks, of course, to the Curiox team, who's been just tremendously helpful in helping us onboard the laminar wash for probably, some kind of non-standard approaches, which required, a whole lot of method development. So we're really appreciative of that. And with that, I'm happy to answer some questions.
Thank you, Rebekah, for your informative presentation. That was wonderful. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. Rebekah, this first question says, the surfaceome maps achieved through the cell surfer platform work superbly well with the discovery of cell surface and glycoproteins at a wide range of less than 0.3 to 10 million cells in under 45 hours, which is a reduction in terms of starting material. Could the platform be further enhanced through sample miniaturization methods or better debris removal?
Sure, I think... Yeah, absolutely. So the kind of the last slide I ended with the nanoscale cell surface capture, I think again, the devil's in the details. Every single step of the process is kind of a painful method optimization in its own. And so for everything up until the most recent, we had really optimized everything on after the cells are labeled, how do we handle them the rest of the way? And so that's kind of figured out. And then, as I mentioned, the laminar wash is really amazing for that front end. So there's a lot of, in our work and a lot of other proteomics sample preparation strategies where you're adding a reagent to your cells, you have to incubate for some amount of time, and then at some point you need to remove that, but you want to keep your cells. And all of those processes are traditionally done with centrifugation. Centrifugation stresses the cells, you lose a ton of material that way. So absolutely, we definitely know that now with swapping out all of those manual steps with the Curiox laminar wash, we're really able to start with small numbers of cells, allowing us to work with, let's say 20 micrograms of material from the start. So this is really opening new doors for us and is really exciting.
Great, thank you. Another question I have for you here. It is interesting that LSMEM2 can act as a useful quality control to assess whether the cell preparation or isolation method preserve the full repertoire of surface proteins. Is there a need for more gentler processes for cell preparation based on your experience?
Yes, I think there's probably lots of other cell types in the human body that are similar. We just haven't worked with them. Our experience with cardiomyocytes, the least amount of sample handling, the better the data. Whether it's proteomics or transcriptomics, we really would like to find ways to, how do you disturb those really stiff extracellular matrix connections between all the different cell types in the heart? How do you, then get rid of everything else other than the cardiomyocytes? It's absolutely very technical. So having strategies, and I'm always interested in alternative enzyme choices, because one of the challenges is that, collagenase 4 from company number 1 is not the same as collagenase 4 from company number 2, even different lots. So they have different amounts of residual tryptic activity, which can be really problematic. Because remember, trypsin doesn't care who you are. It will just cleave you if you have lysines and arginines, where at least collagenase, hopefully, is more restricted to just the collagens. So I think that's really critical. When we think about doing single cell, proteomics of heart cells, if you're using trypsin or other enzymes that their job is to cleave proteins, it's going to be really, impossible to get full coverage of the proteome of that cell type. So absolutely anything we can do from kind of a physical to an enzymatic approach to be gentler on the cells, cause less stress, preserve the cells as much as possible, will only make for better data.
Wonderful, thank you. Now, Rebekah, what is the most challenging task for you as the chief researcher who studied LSMEM2's impact when you tasked this to take this particular biomarkers for clinical evaluation?
Yeah, I think, there's a couple of different things. There's the technical aspect, which I think we have covered. The nice thing about being a mass spectrometry lab is we can develop very highly sensitive, highly specific, custom targeted assays for LSMEM2. So we don't really have to rely on antibody-based. Even though we did make an antibody, we don't have to rely on that for the detection. And the nice thing is, now that we have these custom targeted SureQuant assays, we can apply it to kind of any cell, any tissue, serum, plasma, any biological fluid. So that isn't really so hard. I think the challenge is really getting the right sample cohorts. As probably it's kind of a common theme that if you're working with human, there's a lot of comorbidities to consider. There's a lot of variability in sometimes how samples are collected before they're put in the biobank. There's a lot of technical aspects that you have to control for as much as possible, to make sure that your readout at the end, if you're seeing a trend with cohort, patient group A versus patient group B, is it really related to the utility of this protein to differentiate between two groups, or is it somehow related to some aspect of sample handling? And so those technical challenges are probably some of the bigger things that we think about.
Great. Another question for you here. From your experience, could cell surfer and better sample preparation technologies like laminar wash retrospectively look at clinical data to identify a biomarker?
Like retrospectively, as in data that's already generated? I don't know about that. I think about them using experimentally. I think the data that we have from Cell Surfer in our vision repository is helpful for that, because we find a lot of proteins that have been previously described as being cell type specific really are not.
Thank you. We're getting some great questions in from our audience here. This next question says, it sounds like your established workhorse of sample preparation, sample acquisition, and cell surfer-based analysis have increased sensitivity of detection and may work with more precious sample types. Have you had any experience with other species and organ-specific samples, for example, non-human primates or avian species? And can your platform help drive costs down?
Yes, to all of the above. So we have a lot of interest in exploring different organ-specific samples. So our work with Sally Temple at the Neural Stem Cell Institute in New York, they're working with acutely isolated retinal pigmented epithelial cells from human eyes, tremendously limited sample source. And this technology is working great for that. In terms of other species, we work with rat and mouse pancreas. So we have collaborators that actually do the animal part, but we are able to map proteins on the cell surface from beta cells isolated from pancreatic islets from rats and mice. Really great and informative, but we definitely find a lot of differences between the animal models and the human. And so then again, it's just, it's a prioritization process of we find lots of beta cell specific markers in the rat that are not present in human. This is just, it comes a bioinformatic challenge at the end. And so I think, you know, all those different things are coming. We don't have any collaborations at the moment for non-human primates or avian, but, you know, certainly are interested in exploring that. In terms of helping drive down the cost, you know, it kind of, I don't know that it drives down the cost, because the more fancy we get with instruments that make it faster and easier to do, then we're adding, you know, more costs in terms of, you know, financial costs in terms of, you know, purchasing, operational costs, service contracts, things like that. So I don't know that it will ever be really cheap, but at least we can do it faster, more reproducible, better quality data, more quantitative. So it's beneficial on that end. And then also it enables us to use a lot less starting material. I mean, when I was a postdoc, we would use like 15, 10 centimeter plates of cells to do the cell surface capture. And now we're down into, you know, one well of a 24 well plate. So I think kind of cost just depends on your perspective.
Great, thank you. Next question asks, from a cell-cell synapse point of view, do you have any comments about using this platform to study unique cell-cell potency or cytotoxicity? And could you share your insights on performing those in vitro assays?
Well, I would say from a generic perspective that when we're thinking about cytotoxicity or cell-to-cell potency, the proteins that sit at the surface of the cell control those processes, right? So knowing more about what's available, what receptors are available, absolutely. I think, you know, we think about some drug targets. I got to remember the examples that I have for that. Some drug targets like Cerevastatin was a drug that was proposed for lowering cholesterol, and then there was a TGN 1412 for immunotherapy for cancer. And these are two drugs that made it through preclinical testing, but were later found to cause severe organ failure and kidney failure in the case of Cerevastatin. And the reason the toxicity wasn't found in preclinical testings is because the models that were used did not express the full repertoire of cell surface proteins that are found in the human. So I think TGN 1412 is something that targets CD28 on T cells, and that in the non-human primate model used for testing doesn't actually have CD28 on that subpopulation of T cells. And then I think Cerevastatin, I forget the exact name of the transporter, but it's an anion transporter that is present in humans, wasn't even present in the mouse models used. So absolutely, our hope is that by enabling mapping of surface proteins from primary cells from humans, there will certainly be advantages to doing those in vitro assays on primary human cells and avoiding the challenges we have with cell lines that don't really mimic or recapitulate what's in the human, as well as problems with animal models.
Great, thank you. We have time for just a few more questions here, so we'll ask these and then we'll wrap up shortly. This next question asks, it sounds like there were several limitations between the methodologies of detection via single cell sequencing, flow-based characterization, and mass cytometry. Was there any insightful aspect when targeting cellular differentiation to detect LSMEM2 transcripts reproducibly?
Yeah, I think it comes down to just remembering that when you're looking at a tissue. So I have some data on this that I didn't present, but when we followed up with LSMEM2, and we found these changes in heart failure. So we know that a lot of the data that we have in this paper have not been previously described, which is exciting, but it's also like, why? Is it because we're wrong, or is it because it's really new? Could be lots of things. And so we did ask, all right, so we see a decrease in protein abundance for LSMEM2 in cardiomyocytes when we do cell surface capture. Well, we have to acknowledge that the cell surface capture method is going to be biased towards, it only detects peptides that are glycosylated. So if there's a change in the sugar, somehow that might affect our labeling and our enrichment. And so maybe the change we see in abundance by cell surface capture is really not due to the total protein, but is due instead to the sugar. So then we took whole tissue, left ventricle, failing none, and did qPCR, and we did not find a difference. Well, there's lots of reasons that RNA is not going to predict protein abundance. So that wasn't a big surprise. But then we did our targeted quantitation by mass spectrometry, our SureQuant assay, again on tissue, homogenate, and also did not detect a difference. And it wasn't until we isolated the cardiomyocytes and did SureQuant targeted on that did we again see our trend difference of lower in failing versus none. And I think all of this just reminds us that if you're using a molecular approach, if you're doing a molecular measurement on a sample that is a homogenate of tissue for which there are lots of different things in that tissue. So we have myocytes, endothelial cells, fibroblasts, macrophages, and a ton of extracellular matrix. You can't control for that distribution across samples. So, and you're going to have variability. In some cases, you're going to have more extracellular matrix. So when you do proteomics, for example, and you normalize the total protein, oftentimes what's going to happen is that your subtly changing protein abundances may be more difficult to detect. Same from the transcript level, that, when you homogenize and you're averaging that signal across, and you haven't controlled for the fact, well, maybe this sample has more of this cell type or more extracellular matrix, is really going to cause a problem. And so I think that going forward, it just reminds us of why it's worth all of the effort to isolate the cells. Tissue homogenate is just going to provide a limited view of what's really going on.
Great. Well, thank you so much, Rebekah. We are out of time for today, so we're going to go ahead and wrap up here, but I do want to provide a final thank you again to Rebekah for your time today and your important research. We would also like to thank Labroots and our sponsor, Curiox, for underwriting today's educational webcast. Now, before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.